What if a tiny molecule could save a human life? From the lab bench to the pharmacy shelf, this is the incredible journey of how molecules become medicines. Matter is anything that has mass and occupies space. But in medicinal chemistry, matter becomes far more complex. Drugs are built from atoms, molecules, ions, proteins, solvents, and biological membranes. Yet medicinal chemists don't just see structures. They see electron density, charge distribution, hydrophobic regions, and reactive sites. A drug molecule is not static. It's a dynamic cloud of electrons constantly moving through space, interacting through quantum forces. Understanding this invisible motion is how scientists transform simple molecules into life-saving medicines. Every medicine on Earth is built from atoms. But in medicinal chemistry, each atom has a specific role. Carbon forms the backbone of drug molecules. Hydrogen controls shape and flexibility. Oxygen creates hydrogen bonds. Nitrogen helps drugs bind to receptors. Sulfur affects metabolism, while fluorine increases stability and potency. Chlorine helps drugs cross biological membranes. Take paracetamol as an example. Its carbon framework, oxygen and nitrogen atoms control how the drug dissolves, absorbs, metabolizes, and interacts inside the human body. In medicinal chemistry, changing just one atom can completely change a medicine's power. In medicinal chemistry, shape is everything. A drug only works if its 3D structure perfectly fits a biological target. This is called the lock and key interaction. The drug acts as the key, and the protein receptor acts as the lock. If the molecular geometry is even slightly wrong, the drug can fail. Changing just one atom, one bond angle, or one stereocenter can completely alter biological activity. A famous example is thalidomide. One mirror image form was therapeutic, while the other caused severe birth defects. Electrons control a drug's polarity, reactivity, acidity, hydrogen bonding, and binding strength. When drugs interact with proteins, they use invisible electron-based forces. Hydrogen bonds create attraction between electronegative atoms. Ionic interactions pull positive and negative charges together. Van der Waals forces create tiny temporary attractions. And pi-pi stacking occurs when aromatic rings interact through electron clouds. These microscopic forces decide whether a drug becomes powerful, selective, or toxic. In the end, every medicine works because of how electrons move and interact inside the human body. At the molecular level, matter is always moving. Drug molecules constantly rotate, vibrate, bend, twist, and collide with surrounding water molecules. Even proteins inside the body continuously change shape. This creates something called dynamic binding, where the drug and receptor constantly adjust to each other in real time. Water controls solubility, transport, protein folding, membrane crossing, and bioavailability. If a drug is too hydrophobic, it won't dissolve properly. If it's too hydrophilic, it can't cross biological membranes. That's why medicinal chemists carefully balance water-loving and fat-loving regions inside a molecule. This balance is called lipophilicity, commonly measured using log P and log D. Let's move to next. A drug only binds to a target if the interaction is energetically favorable. Two major forces control this process: enthalpy and entropy. Enthalpy comes from molecular interactions like hydrogen bonds and electrostatic forces. Entropy represents disorder and molecular freedom. At the deepest level, matter follows the laws of quantum mechanics. Electrons do not orbit atoms like planets. They exist as probability clouds, wave functions, and quantum states. Quantum chemistry allows scientists to predict reactivity, molecular orbitals, charge distribution, and transition states inside drug molecules. Today, modern drug discovery combines quantum mechanics, machine learning, and molecular docking to design next-generation medicines faster than ever before. In medicinal chemistry, understanding quantum behavior means understanding how molecules truly interact at the atomic level. Consider imatinib, a revolutionary anti-cancer drug. Medicinal chemists precisely engineered its molecular shape, hydrogen bond donors, aromatic rings, and electronic properties. Why? So the molecule could selectively bind to the BCR-ABL kinase protein responsible for leukemia. Once bound, cancer cell signaling is shut down while healthy cells are affected far less. This breakthrough completely transformed leukemia treatment. The future of medicinal chemistry is becoming fully AI-driven. Next-generation drug discovery will use AI-designed molecules, robotic synthesis, quantum simulations, digital twins of proteins, and generative molecular design. Autonomous laboratories may soon design, synthesize, and test new medicines with minimal human intervention. If you found this helpful, don't forget to like and subscribe for more deep dives into the molecular world. See you in next video. Happy learning chemistry with Harshada.